You ever look at your family and just wonder why? Why do some traits, like your dad's sense of humor or your grandma's curly hair, just dominate for generations, while other things seem to vanish? Well, we're about to explore an idea that is so out there it might just rewrite how you think about life itself. The idea that the answer isn't just in our genes, but in the totally bizarre, spooky rules of quantum physics.
Right, let's get into it. This is one of the most basic questions in biology. We all learned the high school version of genetics, but the deeper you dig, you realize pretty quick that those simple rules, they don't even begin to cover it. There's a real mystery at the heart of why one version of a gene just completely bosses another one around.
And this is where things get really wild. Some incredible new research is suggesting that to actually understand our own biology, we have to shrink our perspective. I mean, way, way down. Past the cells, past the famous double helix of DNA, all the way down to the subatomic level. You know, the place where particles can be in two places at once and literally ghost right through solid walls.
Okay, but before we jump completely down that rabbit hole, let's do a quick refresher on the classical rules. This is the stuff we all know, the Biology 101 that sets the stage for all the quantum weirdness that's about to come.
You definitely remember this from class, right? We get two versions of every gene. They're called alleles, one from each parent. So if you get two different alleles, you're what's called a heterozygote. And in that showdown, the dominant allele is the one that wins. It's the trait you actually see. The other one, the recessive allele, it just gets quiet. It's supposed to be a simple, predictable on-off switch. Or is it?
Yeah, because this is where the simple story starts to fall apart. That clean on-off switch model, it's not the whole picture. It's not even close, actually. So on paper, it's all neat and tidy. One gene wins, the other loses. End of story. But in the real world, it's messy. Dominance isn't always a sure thing. Sometimes a recessive trait pops up when it shouldn't. The strength of a dominant gene can be all over the place from person to person. So the big unanswered question is, what's the hidden force that's messing with this supposedly simple genetic switch?
To find that force, we've got to go on a journey. We're going to zoom in from the scale of you and me all the way down into the quantum foam where reality just gets wonderfully bizarre.
All right, so we're starting here, at our scale, the world of organisms, where things make sense and follow the familiar laws of physics.
Now, let's zoom. We're pushing past all the tissues and cells, diving right into the command center, the nucleus, to find that famous blueprint of life itself, the DNA molecule.
And here we go. The final leap into the world of the unimaginably small, the quantum realm. This is where the actual particles that make up our genes live, and down here, they play by a completely different and, frankly, much weirder set of rules.
So what are these rules? Well, scientists are now zeroing in on at least 3 of these super strange quantum effects that seem to be happening inside our DNA, potentially messing with the traits we inherit. Let's break them down.
First up, we have something called proton tunneling. Now, in our world, if you throw a baseball at a wall, it bounces back, every time. But in the quantum world, a tiny particle like a proton can sometimes just show up on the other side. It tunnels right through an energy barrier it has no business crossing. So check this out. That little ghostly jump can have huge consequences. A proton inside a DNA base can tunnel to a different spot. When it does that, the base temporarily changes shape into a rare form called a tautomer. This little molecular shapeshifter can then accidentally pair up with the wrong partner when your DNA is getting copied. And just like that, you've got a permanent mutation. A brand new genetic instruction born from a quantum leap.
Next, there's DNA charge transport. We usually think of DNA as this giant library, right? A biological hard drive holding all our information. But it turns out it's also an amazing conductor. Electrons can literally zip right along the stacked bases of the double helix, making DNA act kind of like a biological wire.
And the power of this wire is just staggering. Your cells have these tiny protein mechanics that are always crawling along your DNA, looking for damage. Well, it turns out they use this electrical current to talk to each other. And get this, when one of these repair proteins changes its quantum state, its charge specifically, its grip on that DNA wire can get over 550 times stronger. This helps it find and fix mistakes, sure, but it also means that tiny quantum changes can have a massive say in which version of a gene gets repaired and which one is left with a mutation.
And finally, we've got spin chemistry. Every single electron has this property called spin. The easiest way to think about it is like it has a tiny built-in compass needle that's always pointing either up or down. Now, usually these spins are pretty random, but not in our DNA. See, because of its twisted helical shape, what scientists call its chirality, DNA is incredibly picky. As electrons shoot down that biological wire, DNA acts like a bouncer at a club, filtering them and letting one spin direction pass through way more easily than the other. This is called the chiral-induced spin-selectivity effect, or CIS, and the result is wild. You get this massive bias, where up to 85% of the electrons moving through our DNA can end up with their spins all pointing the same way. This quantum bias can then influence everything from DNA repair to how our genes are expressed.
Okay, so we've got tunneling causing mutations, charge transport steering repairs, and spin chemistry filtering electrons. Each one of those is mind-blowing on its own, but what happens when you put all of them together? Well, you get a completely new and honestly revolutionary theory of how our genes work.
It's called Quantum-Modulated Dominance, or QMD for short, and the core idea is pretty simple. Dominance isn't some fixed black and white property of a gene. Instead, it's something dynamic, something that can be turned up or down, tuned, just like you turn the dial on a radio, by all these underlying quantum effects.
Let's see how this all clicks together. Proton tunneling isn't just creating random mutations. It might be biased toward creating new, more powerful versions of genes. Charge and spin transport aren't just fixing DNA randomly. They could be favoring one parent's version of a gene over the other, essentially helping it survive. And other quantum effects can tweak how loud a gene is, which might be the difference in whether one copy is strong enough to do the job on its own.
So, if our entire genetic inheritance is constantly being fiddled with and tuned by the quantum world, what on earth does that mean for us, for the future? The implications are just huge.
I mean, think about it. This could mean that evolution isn't totally random after all. There might be a quantum thumb on the scale, biasing the kinds of mutations that pop up. For medicine, since things like electron spin are sensitive to magnetic fields, maybe we could one day use electromagnetism to literally tune our own genes up or down. But maybe the biggest thing is this. Our genetic code isn't a fixed blueprint. It's more like a dynamic, probabilistic script that's being constantly edited by the quantum universe.
And that leaves us with one last, absolutely mind-bending question to chew on. We've always thought of life as this incredibly complex but ultimately predictable machine. But what if its most fundamental rules are written in the language of quantum probability, of tunneling ghosts and spinning compasses? What does that say about our own destiny? Is life, at its very core, just as weird and unpredictable as the quantum world it's built from?
Welcome back to the deep dive. For centuries, really, the bedrock of genetics has been Mendelian inheritance. You know, those simple rules deciding why you might have your dad's eyes but your mom's hair.
Right, the classics.
Today, though, we're taking one of the oldest ideas in that playbook, genetic dominance, and looking at it through a lens that's, well, pretty radical. Quantum physics.
It sounds almost sci-fi, doesn't it? But think about it. If you're a heterozygote, you got different gene versions, alleles, from each parent, say big A and little a. Why does the big A often completely mask the little a?
That's dominance, right? The A dominates A.
Exactly. And we actually quantify that strength. There's a term, the dominance coefficient. It tells us how strong that masking effect is.
And traditionally, we've explained allers using pretty standard biological ideas. Things like gene dosage, how much protein gets made, is half the amount enough?
Yeah, or haploinsufficiency, where half just isn't enough. Plus regular feedback loops in the cell. It's all been about biochemistry, really. Amounts. Concentrations.
But the source material we're diving into today, this big synthesis report on something called Quantum-Modulated Dominance, or QMD, it argues we've been missing a huge piece of the puzzle.
It really turns things on their head. The core idea is that this super precise molecular events setting, which allele gets used, things like protons jumping, electrons moving, enzymes working, these aren't just following quantum rules passively.
They're being actively shaped by them.
Precisely. Actively modulated. These quantum effects, things like tunneling and spin, they introduce a bias. They're constantly tweaking that dominance coefficient all or dollars. It's not just fixed chemistry.
Okay, that's a lot to unpack. So over the next, say, 25 minutes or so, that's our mission. A deep dive into the evidence. We're looking at research across 6 different areas, showing how the subatomic world messes with inheritance.
Yep. We'll look at 3 key stages. How mutations happen, how they get repaired, and how genes actually get expressed. And we're not just hand-waving here.
No, we've got specific data points, things like kinetic isotope effects, spin polarization percentages, real numbers that suggest the old classical rules aren't the whole story.
And for you, the listener, think of this as a shortcut.
You don't need the PhD in quantum chemistry, but you can grasp this core idea.
Yeah.
Your own gene expression might be influenced by factors you'd never think connect to your DNA.
We're hoping for some real aha moments about why you express the genes you do. Surprising stuff.
Definitely non-intuitive.
All right, let's start right at the source. Mutation. The textbook picture is usually random errors, right? Like typos during DNA copying, mostly due to thermal jiggling.
Random thermal noise, yeah. That's the standard view.
But QMD says, hold on, there might be a quantum bias baked into how often and where these errors pop up. And the mechanism. Proton tunneling.
Okay, so picture the DNA double helix. You've got base pairs holding the strands together, like guanine G paired with cytosine C. They're linked by hydrogen bonds.
Involving protons.
Exactly. And protons are tiny, very quantum particles. Now, classically, for a proton in that GC bond to hop over to a slightly different spot, which can cause problems, tills, it needs enough energy to get over an energy barrier. Think rolling a ball uphill.
Needs a push.
Needs energy. But quantum mechanics allows something weird for small particles like protons. They don't have to go over the hill.
They can tunnel through it.
They can tunnel right through it. It's a wave thing, a probability game.
Yeah.
And sophisticated models, these open quantum systems models, they show this tunneling boosts the rate of protons making that jump by orders of magnitude compared to just classical thermal hopping.
Orders of magnitude, so not just a little bit faster.
Way, way faster. It makes an event that would be incredibly rare, much more likely.
And when that proton tunnels shifts position within the base pair, what happens then?
It creates something called a tautomer. The chemical formula is the same, but the atoms are arranged differently, specifically the hydrogen atoms, and this changes everything for replication.
How so?
Because the tautomer, let's say GC, it doesn't pair correctly anymore. When the DNA copying machinery, polymerase, comes along, it sees this weird GC and might grab the wrong base to pair with it, like putting a thymine T where a cytosine C should go.
Ah, so that's the mistake.
That's the initial mistake. And then in the next round of copying, that T gets paired with an A, and boom, you've locked in a mutation. A GC pair has become an AT pair.
That's called a transition mutation.
Exactly. The most common type of point mutation, these subtle swaps. Now, the numbers here are interesting. The source estimates the chance of a base pair being in that tautomeric state at any given moment is tiny, around $1.73 times 10 to the 4 body temperature.
Pretty rare.
Seems rare. But DNA replication happens billions and billions of times. And the quantum tunneling effect dramatically increases the rate at which bases can flicker into that tautomeric state. So even with a low probability of being in the state, the high rate of getting there means this mechanism introduces a subtle but consistent bias into the whole mutation process.
Okay, follow the thread. How does a biased mutation spectrum end up affecting dominance, the Dollar coefficient?
It's about evolution playing with loaded dice. If tunneling makes certain transition mutations more likely, and these mutations happen to occur in places that, say, make a protein work better or get overexpressed.
A gain of function allele?
Precisely. If the quantum bias favors creating these gain of function alleles, which often act dominantly.
Because even one copy making lots of functional product is usually enough to see the effect.
Right. Then the quantum tunneling, way down at the subatomic level, is essentially biasing evolution towards creating more dominant alleles over long timescales. This would naturally push the average dominance coefficient higher in a population.
And this isn't just theory. You mentioned we can measure this quantum influence.
Yeah, there are two key experimental tools. The tunneling to classical rate ratio, which we call kappa, and the kinetic isotope effect, or Ki.
Ki sounds important.
Ki is the smoking gun, really. It's clever. You compare the speed of a reaction using normal hydrogen with the speed using deuterium, which is heavy hydrogen, same chemistry, just twice the mass.
So it's harder for deuterium to tunnel.
Much harder. Mass matters hugely for tunneling. If swapping H for D dramatically slows the reaction down, tunneling was a critical rate-limiting step. And the data for some of these DNA misbearings like GT wobbles show Ki IE values up around 10.
10. And you said anything over 2 usually confirms tunneling.
Yeah, 10.15 in one case.
Yeah.
It's massive confirmation. Tunneling is definitely happening.
And the kappa ratio, what does that tell us?
That tells you how much faster tunneling is. For that GT wobble misincorporation pathway, calculations found a kappa is around 99.
99 times faster.
Almost 100 times faster than the classical thermal way of making that mistake. This quantum pathway just dominates for that specific error. It massively amplifies these misincorporation events, pushing mutations towards GC to AT changes. It's physics biasing genetics.
That's Yeah, that's pretty staggering. But we need balance here. The report mentions a counter-argument, right? The E. coli experiment with heavy water.
Absolutely crucial piece of the puzzle. If tunneling is so potent, you'd expect that growing bacteria in heavy water, D2O instead of H2O, would drastically slow down all proton tunneling everywhere and slash the overall mutation rate.
Makes sense. What happened?
They did find a decrease, but it was small, only about a 0.663-fold change, so roughly a 34% reduction, and critically, it wasn't statistically significant.
So massive effect in specific reactions, but barely noticeable across the whole genome. How do we square that?
It doesn't mean quantum effects are irrelevant. It strongly suggests they're highly localized. The tunneling effect likely depends intensely on the exact shape of the energy barrier, which is determined by the local DNA sequence, the neighboring bases, how they stack.
So some sequences might have barriers perfect for tunneling, others not so much?
Exactly. Tunneling might dominate in specific hotspots while classical effects rule elsewhere. But those hotspots could be precisely where mutations leading to dominant alleles are most likely to form. So QMD isn't saying quantum effects rewrite everything everywhere. It's saying they modulate things in specific, sequence-dependent ways. It's targeted bias, not a global change.
Okay, so quantum mechanics can help create errors with a bias. Now let's flip the coin. How does it influence fixing those errors? And why might that fixing process also be biased towards one allele?
Right, we're moving from the proton to the electron now, and looking at how electrons move through DNA. This involves DNA charge transport, or CT.
I thought DNA was mostly an insulator.
That was the old thinking for decades. But it turns out the way the base pairs stack up, like a pile of coins, allows electrons to hop or flow along the helix quite efficiently under certain conditions. It basically turns DNA into a tiny wire.
A wire? What's the current used for?
Two key things. It allows oxidative damage from reactive oxygen species to travel long distances down the DNA. But crucially for this discussion, repair enzymes use it as a super-fast scanning mechanism.
How does that work? They send a little pulse down the wire?
Sort of. Think of it more like checking conductivity. Healthy DNA conducts charge well, but a lesion, a damaged base, a mismatch or break, acts like a roadblock. It stops the charge flow.
Like a blown fuse.
Exactly. And the efficiency of this wire is very sequence-dependent. Long stretches of purines, the A's and G's, are great conductors. Pyrimidines, C's and T's, not so much. And any damage really kills the conductance right there.
So repair enzymes can feel for these roadblocks electronically, faster than physically checking every base.
Much faster. This brings us to a really cool example. Proteins with 4Fe4S clusters. These are iron-sulfur clusters, like little redox batteries inside the protein.
Redox meaning they can gain or lose electrons?
Right. And proteins like DNA primus, endonuclease III, muti-key repair enzymes use these clusters in DNA charge transport to find damage. The whole thing hinges on the cluster's electrical state, which dramatically changes how tightly the protein binds to the DNA.
How dramatic are we talking?
Okay, get this. When the iron-sulfur cluster in DNA primus gets oxidized, loses an electron, going from 4Fa4S2 plus to 4Fe4S3 plus its grip on the DNA, its binding affinity increases by at least 550 times.
550-fold? That's huge!
It's the difference between loosely scanning and being glued to the spot. And this switch is triggered by DNA binding itself. The potential shifts by about 200 millivolts.
So how does the charge transport use this switch to find a lesion?
It's elegant. When the protein, in its high-affinity 3+ state, binds to healthy DNA, charge flows through the DNA wire to the protein's cluster. This reduces the cluster back to the 2+ state.
The low affinity state.
Right, so the protein lets go and slides along, ready to check the next section. But if it binds near a lesion...
The charge transport is blocked. No current flows.
Exactly. The protein stays stuck in its oxidized, high-affinity 3+ state, trapped right at the site of the damage, flagging it for repair.
Okay, I see the mechanism. Now, connected back to dominance, to our dollars, how does this create an allelic bias?
It comes back to sequence dependence. Remember, CT efficiency varies with the sequence. Imagine our heterozygote again, big A, little a. Maybe allele A has a sequence near a potential damage site that's a fantastic conductor, lots of G's and A's. Allele A, same spot, maybe has a CT-rich sequence, a poor conductor.
So the electronic scanning works better on allele A?
Potentially, yes. The repair enzyme might detect damage more quickly or efficiently on allele A because the charge transport signal works better. Or perhaps the trapping mechanism is more sensitive there. If allele A gets repaired more reliably or faster than allele A over time.
The allele A survives better. Its functional product is more consistently available.
Precisely. It gets preferential treatment just because of its physical sequence's electrical properties. This biases the outcome in the heterozygote, nudging dollars towards dominance for allele A. So the QMD model needs a repair bias factor, shrew of dollars incorporating that redox shift, delta E.
And there's another layer to this electron story, isn't there? Something even stranger involving spin.
Yes. If charge transport wasn't weird enough, we have chiral-induced spin selectivity, or CISS. This adds magnetism to the mix.
Okay, chiral means handedness, right? Like DNA being a right-handed helix.
Exactly. It has a specific twist. CISS is the phenomenon where, as electrons move through this chiral twisted structure, the DNA helix acts like a filter, preferentially allowing electrons of one spin, say spin up, to pass through more easily than the other, spin down.
DNA is a spin filter.
Yes.
Seriously.
The electron spin interacts with the helix's chirality, and the effect can be incredibly strong. Experiments on single DNA molecules have shown spin polarization, the preference for one spin over the other reaching up to 85%.
85%. That's almost perfect filtering. But that sounds like a lab result. Does that kind of extreme effect really happen inside a warm, messy cell nucleus?
That is the critical question. We know CISS happens. We see high polarization in controlled settings. The big unknown is the magnitude in vivo. But even if it's significantly less than 85% in the cell, any substantial spin polarization means electron transport in DNA isn't just about charge, it's about spin.
And why does spin matter for repair or expression?
Because many biochemical reactions, especially redox reactions involving electron transfer, are spin-dependent. If the electrons arriving at a repair enzyme via DNA are already spin-polarized by CISS, that could influence the enzyme's activity or efficiency in a way that depends on the specific DNA sequence and its CISS properties.
So the physical structure of the allele dictates not just charge flow, but spin-polarized charge flow, another layer of sequence-dependent bias.
Exactly. So that repair bias factor, dark dollars, needs to include not just the redox shift, delta E, but also the spin polarization term, part of it. It's quantifying how an allele's electrical and magnetic properties influence its survival.
Okay, we've biased the mutations coming in, biased the repairs keeping alleles alive. Now for the final step, expressing the gene. Getting the protein made. QMD argues quantum effects set the actual on-off threshold here too. This is quantum epigenetics.
Right. Epigenetics is all about controlling gene access, adding or removing chemical tags like methyl groups on DNA or the histone proteins DNA wraps around. These tags act like switches.
And enzymes flip these switches.
Enzymes flip the switches. And just like proton tunneling messed with mutation rates, tunneling can affect the speed of these epigenetic enzymes. And that speed determines whether a gene crosses the threshold to be on.
So the enzyme's reaction rate isn't purely classical chemistry?
Not entirely. Yeah. Again, we look for the Chi-E, the kinetic isotope effect. Take LSD1 demethylase. This enzyme removes metal groups from histones, generally activating genes. Experiments show a Chi of about 3.1 for its key chemical steps.
3.1. That's solid evidence for tunneling again. What's tunneling this time?
It's likely a hydride ion, a proton with 2 electrons. So the rate at which this important gene activator works is fundamentally limited by a quantum tunneling event.
And it's not just LSD1.
No, another big one is DNMT1 methyltransferase. This is the enzyme that adds methyl groups to DNA, usually silencing genes. Very detailed Chi-E studies using multiple isotopes, heavy hydrogen, carbon-13, even sulfur-36 conserved quantum corrections were needed to explain its reaction rate. They could practically map the transition state atom by atom using these quantum effects.
So quantum mechanics is setting the tempo for the epigenetic machinery. How does that tweak the dominance coefficient to dollars?
It comes back to thresholds. Let's think about a haploinsufficient gene again. Normally recessive because 50% protein product isn't enough.
Right, you need both alleles working full tilt.
Okay, but what if the enzyme needed to activate that gene or remove a silencing mark is slowed down because its reaction relies on tunneling? If there's a high Chi-E, the enzyme works slower.
So the total amount of protein produced, even from the good allele, might dip below that critical functional threshold.
Exactly. The quantum slowness, the high Chi-E, effectively raises the bar for expression. It makes it harder for the gene to reach the on state or produce enough product. This can push a gene that might have been borderline or even partially dominant firmly into the recessive category. It lowers the dollars.
So the QMD model needs an epigenetic correction factor, equalize, based on these Chi-E values.
That's right. It quantifies how quantum tunneling in enzyme catalysis shifts the functional output.
We've dealt with internal quantum effects tunneling inside DNA, inside enzymes. Now for something really wild, an external quantum influence. The magnetosensitive gene switch and cryptochrome.
Yeah, this is where it gets really interesting, linking the environment to the genome via quantum mechanics. Cryptochromes, CRY, are proteins found pretty much everywhere, from plants to us. They're key players in the circadian clock, our internal timekeeper.
How do they work?
When light hits cryptochrome, it triggers a reaction that creates radical pairs. These are basically two molecules that temporarily have unpaired electrons. Their fate, what happens next chemically, depends crucially on the spin state of those two unpaired electrons. Are they aligned triplet state or anti-aligned singlet state?
Okay, electron spin again. But what's the magnetic connection?
Here's the amazing part. The relative energies of the singlet and triplet states and the rate they interconvert are incredibly sensitive to external magnetic fields, even really weak ones.
Like the Earth's magnetic field.
Exactly. Think of the radical pair like a tiny compass needle flickering between states. The Earth's magnetic field provides a very subtle alignment force. Experiments show that the yield of the chemical reaction in cryptochrome can change by 10–20% just by applying a magnetic field of about 28 millitesla.
Okay, 28 millit is maybe lab strength. What about the Earth's field, which is much weaker?
That's the kicker. The effect is still significant, measurable, down at fields around 50 microtesla, which is right in the range of the Earth's geomagnetic field.
Wait, so the planet's magnetic field can directly influence a chemical reaction inside our cells that regulates our daily rhythms?
That's what the evidence strongly suggests. It provides a direct physical link between the environment and the core gene regulatory machinery.
And can we? Can we fiddle with this ourselves?
We can. Researchers have shown you can use applied radio frequency, RF fields like radio waves, to specifically interact with the electron spins in those radical pairs. You can essentially tune the outcome of the reaction by manipulating the spin states with RF.
Okay, bringing it back to dominance, how does my proximity to a magnetic field or an RF source affect allele A versus allele A?
If the expression of, say, allele A is tied into this cryptochrome pathway, maybe controlling its timing or level, then changing the magnetic environment could subtly alter allele A's output relative to allele A.
So the dominance isn't fixed? It could fluctuate depending on the magnetic fields I'm exposed to.
That's the implication. Dominance becomes a dynamic property, modulated not just by internal quantum kinetics, but also by external physical fields acting on quantum spins. It makes dollars potentially sensitive to the environment in a completely non-classical way.
Which explains why QMD aims to unify all these effects, tunneling, charge transport, spin into one framework.
Exactly. It's trying to capture the sum total of these quantum modulations on the final phenotype.
All right, we've seen the pieces. Quantum effects, biasing, mutation, repair, and expression. How does the QMD theory actually put these together mathematically to predict that shift in the dominance coefficient dollars?
It builds on the standard model, the classical dosage threshold ideas. They aren't wrong, just incomplete. QMD essentially says, okay, that rate at which allele A produces its product, let's call it K dollars. We need to replace that with a quantum-corrected rate, ticks to quantum.
A rate that reflects all these probabilistic quantum events we've been talking about?
Exactly. And this Kaplan text is built up using correction factors that represent the three stages we discussed. We need parameters that capture the quantum influence at each stage.
Let's recap those factors. What goes into each one?
Okay, first, the mutation bias factor. This deals with the input, how likely is a dominant acting allele to arise or persist due to mutation bias. It incorporates kappa, that tunneling to classical rate ratio.
How much faster tunneling makes mutations happen?
Right, and also the probability of being in that mutation-prone tautomeric state. CalR captures the quantum bias on the source code itself.
Got it. Factor one, mutation bias. What's second?
Second is the repair bias factor. This is about maintenance. How likely is an allele to survive damage based on its quantum properties? This factor includes delta E, the redox potential shift in those 4Fe4S repair enzymes.
How effectively they get trapped at damage sites based on charge transport.
Yep. And it also includes just the spin polarization from the CISS effect. My collar basically quantifies if allele A gets better quantum policing than allele A because of its sequence's electrical and magnetic properties.
Okay. Mutation bias, repair bias. What's the third factor?
The third is the epigenetic correction, This This tackles the output stage expression level. It's primarily driven by the key A values measured for those key epigenetic enzymes like LST1 and DNMT1.
Quantifying how much quantum tunneling slows down or speeds up the gene activation or silencing machinery?
Exactly. ECAL reflects how quantum kinetics at the enzyme level can shift the effective expression threshold for an allele.
So you plug these quantum parameters, katika, delta A, Ki, As, LE, into the model. What does it predict happens to the alleles?
The simulations show clear trends. If you increase the factors that favor creating or maintaining a functional allele specifically, higher tunneling rates causing gain-of-function mutations, or better repair via charge bin transport, delta APSLA, the model predicts double increases. The allele becomes more dominant.
Makes intuitive sense. What about the enzyme tunneling, KIE?
That works the other way. If Ke is significantly greater than 1, meaning tunneling slows the enzyme down, it acts like a brake on expression. This tends to make genes, especially haploinsufficient ones, behave more recessively. It lowers dollars.
And the overall potential shift, is this just fine-tuning or can it really flip dominance?
The model suggests it's not just marginal. The combined effect of these quantum parameters appears capable of shifting non-allele dollars across its entire theoretical range from 0 assessed, completely recessive, all the way to 1, completely dominant. QMD proposes a mechanism powerful enough to fundamentally determine the Mendelian outcome.
It's a compelling mathematical story, but physics models need experimental proof, especially in biology. The report outlines some specific ways to test QMD in living systems, right?
Yes, and this is crucial. Moving from calculation to observable biology, they propose 4 main experimental avenues.
Let's walk through them. First up, yeast. Why yeast?
Yeast is easy to manipulate genetically, and it has cryptochrome-like proteins. The idea is to engineer yeast, where a reporter gene's output depends on CRY activity. Make it heterozygous, AA. Then hit it with weak magnetic fields or specific RF pulses.
The external quantum tuner.
Exactly. If QMD is right, just changing the magnetic environment should measurably shift the relative expression of allele A versus allele A. You'd see dominance change in response to the field.
Okay, second system. Zebrafish embryos. What's the angle there?
Zebrafish embryos are transparent, great for watching development unfold. Here, the focus is on tunneling and repair. They'd use zebrafish with known mutations in those 4FE, 4S repair genes, and they'd raise some embryos in normal water, others in heavy water, deuterated water.
Heavy water slows tunneling.
Right. It should slow proton tunneling in DNA bases and potentially hydride tunneling in the repair enzymes themselves. The prediction is that slowing these quantum steps will worsen the effects of the repair mutation, leading to more severe developmental problems or changes in how strongly a trait shows up, phenotype penetrance. You directly link changing a quantum rate to a visible outcome.
Third, getting closer to us. Human organoids.
This brings it towards potential medical relevance. Take human cells. Use CRISPR to create heterozygotes for known dominant genetic disorders, maybe something like FGFR3 mutations causing certain types of dwarfism. Grow these cells as mini organs, organoids, and then culture them under different quantum conditions. Again, deuterated, medium to slow tunneling, or maybe carefully controlled magnetic fields to influence spin effects. The question is, can we measurably alter the penetrance of the disease phenotype in the organoid just by tweaking the quantum environment? Could we make a dominant disease allele behave recessively?
That would be huge. And the final proposed system.
Cryptochrome null mice. This is about isolating the magnetic effect. You compare normal mice, wild type, with mice genetically engineered to completely lack cryptochrome.
The protein sensitive to magnetic fields.
Right. You expose both groups to either the normal Earth magnetic field or a shielded environment with near zero field. Then you compare gene expression profiles. If specific genes change expression in the normal mice depending on the magnetic field, but not in the CRY null mice.
Then you've pinned the effect squarely on the cryptochrome spin chemistry mechanism.
Exactly. It would provide very strong evidence that this quantum magnetic sense is actively modulating gene regulation in mammals.
It's a solid roadmap for testing. But what are the big unknowns right now? What data do we need most urgently to firm up the QMD model?
2 main things, I'd say. First, we need way more systematic data correlating quantum properties with genetics. We have Chi-Es for some enzymes, CISS data for some sequences, but it's patchy. We need large data sets. Measure Chi-Es for hundreds of key enzymes, map CT and CISS efficiency across diverse DNA sequences, and then see how those quantum numbers correlate with whether genes are typically dominant or recessive.
Connect the physics parameters directly to the genetic outcomes on a large scale.
Yes. And the second big gap, maybe the most critical, is the CISS link. We see that huge 85% spin polarization in DNA fragments. But we need the smoking gun experiment showing that this spin-polarized current coming off the DNA directly influences the activity of something important in the nucleus, like a transcription factor binding or another regulatory protein switching on or off. We need to connect the spin filter to the final decision on gene expression. #outro.
This has been, well, frankly, mind-bending, taking something as fundamental as genetic dominance and seeing it potentially underpinned by these subtle probabilistic quantum effects. So boiling it all down, what's the absolute core takeaway here?
I think the core insight is that dominance isn't just about how much stuff a gene makes. It's an emergent property arising from this interplay of quantum phenomena. It's a statistical outcome shaped by proton tunneling, biasing the mutations, charge and spin transport biasing the repairs, and enzyme tunneling and spin chemistry setting the expression levels. Quantum probabilities are woven into the fabric of inheritance.
And the implications ripple outwards from there for basic evolutionary biology.
Yeah, these quantum biases, even if small in each generation, could accumulate over millions of years, subtly steering the path of evolution, influencing which mutations stick around. It adds a whole new physical layer to the mutation selection balance.
And for medicine, you touched on the organoid experiments.
The therapeutic potential is, well, potentially transformative down the line. If dot dollars can be modulated by the quantum environment, could we develop quantum therapies? Maybe drugs designed to interact with electron spin, or specific deuterated compounds to selectively slow tunneling in a faulty pathway. Could we even use targeted magnetic or RF fields in some cases? It opens up possibilities for treating dominant genetic disorders by essentially tuning the underlying physics to make the faulty allele behave recessively.
It's a radical thought. Which brings us to our final provocative question for you, the listener. We tend to think of heredity as somewhat fixed, deterministic. You get your genes, the code is written.
Right, A, T, G, C, set in stone.
But if QMD is right, if the expression of that code, the dominance, can be dynamically tuned by things like the Earth's magnetic field, or maybe even ambient radio frequencies acting on quantum spin states, does that blur the line between genetic destiny and environmental influence in a way we've never considered?
It really does. If the quantum world is constantly nudging your gene expression, influencing which allele wins out moment by moment, then maybe you aren't just the sum of the genes your parents gave you. Maybe, in a very real physical sense, you also need to consider the magnetic fields around you, the RF environment, maybe even that cell tower down the street, when you think about how your own genes are actually behaving right now.
The environment isn't just backdrop. It might be actively tuning your DNA's quantum dials. A fascinating place to leave it. Thank you for joining us on The Deep Dive.